Okay, hello all. This will be a kind of practice presentation for me to figure out where I still have some weaknesses. In this particular presentation, because I will be presenting this, if everything goes well with the travels, at the NK 2025 conference in Tasmania in actually end of this week or beginning of next week. Um, Feel free to join me for this ride, and then we let me know what you think about the presentation. Okay, so today I will be talking about peptide-specific recognition of viruses and virus strains by natural killer cells. My name is Quirin. I'm an assistant professor at the Center for Infectious Medicine at the Karolinska Institute. And I think it's well established that HLA-E acts as a ligand for the inhibitory receptor NKG2A, but this interaction is dependent on peptides that are presented by HLA. So if an NK cell here in orange encounters a healthy cell in gray in this scenario, the healthy cell presents self-peptides, which then form complexes with HLA-E, and these complexes can interact with the inhibitory receptor NKG2A, instructing inhibition in NK cells and thereby maintaining tolerance against healthy cells. In the case of virus infections, it can happen that upregulation of ligands for activating receptors, here in red, coincides with absence of self-peptides, which then, due to the peptide dependency of this axis, leads to a loss of inhibition and facilitates activation of NK cells against these virally infected cells. And some viruses, such as cytomegalovirus, they provide their own viral peptides, which are then presented on HLA-E and can instill inhibition in NK cells with the goal of mediating immune escape. And so with that in mind, that peptides presented by HLA-E are very important in regulating NK cell activity, also in cancer and in virus infections. With that in mind, we were really interested when SARS-CoV-2 first emerged, whether it could contain peptides that can influence NK cell responses. And first we performed an in silico prediction, and we obtained 3 promising candidate peptides. We tested these candidate peptides with an in vitro cellular presentation assay where we use a cell line that is transfected to express HLA-E, but and it doesn't have intrinsic peptides, and due to the peptide dependency, HLA-E then stays intracellular, and we can add synthetic peptide extrinsically. And only if this peptide is presented on HLA-E, then HLA-E peptide complexes are formed that we can detect on the cell surface. And as you can see here, with one of the 3 candidates that were predicted, we really detected very robust HLA-E surface stabilization here with this NSP13 peptide, even higher than the positive control, which is a well-known peptide from the signal sequence of HLA-C. And now behind my my mini video here at the bottom right, I actually have a link to a QR code to a study where we have looked a little bit deeper into a candidate that was predicted, but then turned out to be a false positive in our hands. That's a peptide sequence from the spike protein of SARS-CoV-2, but I can add the link in the description as well. But coming back to this part, these cellular presentation assays imply to us that in particular this NSP13 peptide is on the one hand HLA restricted, which then means that on the other hand that it could act as a ligand to modulate NK cell responses. And to explore the functional consequences of this, we first employed recombinant receptors. So we loaded cells with peptides and then tested whether these peptide complexes bind to recombinant NKG2A CD94 receptor heterodimers. As you can see here, if we do not add an appropriate peptide, the cells, as I mentioned before, they do not have HLE on the surface and they are not bound by these recombinant receptors. If we load the cells with the self-peptide, these form complexes with HLA-E, so virtually all of the cells are HLA-E positive, and the vast majority of the cells also bind to the inhibitory receptor as expected using the self-peptide. However, when we load the cells with the NSP13 viral peptide from SARS-CoV-2, we indeed see again that HLA-E is stabilized on the surface, that HLA-E is presenting this peptide, but there's really no binding of the recombinant receptor at all. Since this was a little bit surprising, we then applied kind of the reciprocal approach, and we obtained HLA-E tetramers refolded with different peptides from the NIH tetramer core facility, and then we used these tetramers as reagents to incubate with primary NK cells. And as you can see here, this is controlled without adding a tetramer. These NK cells are a mix of cells that express NKG2A and cells that do not express the receptor. And again, if we use tetramers refolded with the self-peptide, we can see that all of the NK cells that express the receptor bind to this tetramer that acts as a ligand, whereas the cells that do not have the receptor, they do not bind. Again here, when using the viral peptide, we did not see any interaction between HLA-E peptide complexes and the inhibitor receptor, implying again that the interaction of this viral peptide presented by HLA-E with NKG2A is at best of poor quality. We also directly interrogated the functional response of NK cells by co-culturing NK cells from healthy donors with peptide-loaded K562 cells. And what you can see here is that on the far right here, if we do not load a relevant peptide onto these cells, these are leukemia cells or NK cells degranulate, as we can detect with CD107 surface mobilization, and they also express cytokines such as tumor necrosis factor. If we load these cells with a self-peptide, then that leads to a complete inhibition of the NK cell responses, since that kind of instructs self and tolerance. And then when we loaded the target cells with the viral peptide in line with what we had seen on the previous data, this still allowed NK cells to degranulate and express cytokines, which is consistent across several donors, demonstrating that presentation of this NSP13 viral peptide does not protect these cells against NKG2A positive NK cell responses. So these data so far suggested presentation of the viral peptide by HLA-E antagonizes inhibition of NKG2A, and through this may facilitate NK cell activation and NK cell responses against this virus. So in a broader sense, this NSP13 peptide could be considered a disadvantage or an Achilles heel for the virus, really prompting the question whether this peptide should be a target for viral evolution. And monitoring viral evolution of SARS-CoV-2 is actually quite straightforward due to the intense global genomic surveillance that is ongoing. And we can visualize viral evolution, for example, with a plot like this, where we can see that at the end of the year 2022, there was really kind of a shift in variants that were prevalent. So at the time very prevalent Omicron BA.5 variant was really taken over and dominated by a subsequent variant here in red, the Omicron BQ1 variant. And interestingly, this dominant shift between the variants or strains coincided with a mutation in the NSP13 peptide sequence. And what really struck our interest was that this mutation that appeared here was located at position 2, and position 2 of such peptides is a key anchor position for presentation on HLA-E. So we kind of employed our established toolbox to really in-depth delineate the features of this mutated peptide or epitope. Using cellular presentation assays comparing the ancestral and the mutated peptide, we could clearly show that the peptide presentation efficiency was diminished. And in addition to this, doing an in vitro infection with either the ancestral wild-type SARS-CoV-2 strain or the BQ1 variant that we obtained from Institut Pasteur, we could observe that the BQ1 variant, so the variant that contains the mutated peptide, showed a lower level of HLA surface expression after infection in vitro. In addition to that, in a collaboration with the structural biologist Adnan Ahur here from Stockholm, they could actually resolve the crystal structure of the two HLA-E peptide complexes. And in comparison, what Adnan and his group could detect here is that the branching of the isoleucine that replaced the methionine in position 2 changed and altered the B pocket occupancy of the peptide in the HLA-E binding groove, further supporting reduced complex stability. And one question that we were really interested in is what are now the functional consequences of this impaired presentation for the NK cell responses against these virus variants. And for this, we kind of extended our established models where we now loaded cells either with pure self-peptide or with a mix of self and viral peptide to model infections. So here we have a mix of self and the BA5 or ancestral, the unmutated SARS-CoV-2 peptide, and a mix of self with the mutated peptide. And then again, we can test whether and to which degree recombinant NKG2A receptors bind to the peptide presenting cells. And here on the right, you can see that these experiments are performed in a setting where all of these conditions have very similar total HLA-E surface levels. So here we attempt to not compare the quantity, the magnitude of presentation, but the quality in a mix of presentation. And on the right, you can see as in our previous data, in line with what we had previously shown, presentation of self-peptides here in orange leads to high binding of the inhibitory receptor. If it's a mix of self and the original, the ancestral peptide, that antagonizes inhibition and reduces binding of the inhibitory receptor. And using a mix of self and the mutated peptide correlates with a recovered binding of this inhibitory receptor. We also performed direct co-cultures of peptide-loaded cells with NK cells from healthy donors. And again, we set this system up using mixes of peptides where the total amount of HLA-E, again, was very, very similar to mainly look at qualitative differences. Here we can once again see that without any peptide, NK cells degranulate and perform missing self responses against these cells. This can be completely inhibited and shut down by presentation of self-peptides. The mix of self and the original ancestral peptide here in blue that allows for NK cell responses due to antagonized inhibition, and the mix of self-peptide with the mutated viral peptide here in red correlates with recovered NK cell inhibition. This is likely due to a restored availability of self-peptides within this HLA-E peptide pool on the targets. So I think, if this is really the case, and if this disadvantage or this Achilles heel is reverted, does this then mean that the virus gains an advantage through this mutation? And I think that's a very good question to ask, but also kind of complicated to address. And we, to address this question, we used a method that was put forward by Jesse Bloom and Richard Nea, where they estimate viral fitness. And this method is based on actually real-world data from 6 million patients that have been infected with SARS-CoV-2, and then their SARS-CoV-2 genome was sequenced. And the estimation of viral fitness works in a way that one compares the occurrence of mutations with their anticipated frequency of these mutations based on the neutral mutation rate of SARS-CoV-2. And the intuition behind this is that if a mutation occurs rarely, then this relates to a poor fitness, while if a mutation occurs very commonly, then this suggests that the virus could prevail, or this variant could prevail, and suggesting superior fitness in resulting in infection of many humans. So in this heat map, what you can see here on the y-axis, that is the amino acid sequence of the ancestral NSP13 peptide. And on the x-axis, you can see all the mutations that were detected that led to an amino acid substitution. And first of all, what you can see is that many of these mutations, or most of these mutations, really resulted in a reduced and a deteriorated viral fitness, here indicated by blue color. And only 2 mutations actually enhanced the estimated viral fitness values. And the one mutation up here that really had the highest enhancement of viral fitness, that's exactly the mutation where position 2, the methionine, is replaced into an isoleucine. So exactly the mutation that we had been studying. So this really suggests that this substitution from methionine to isoleucine is associated with enhanced estimated viral fitness across the entire spectrum of SARS-CoV-2, and not only restricted to the direct competition between the BA5 and the BQ1 variants. So to sum this up, I think collectively our data really further highlights the peptide dependency and specificity of the HLA-E-NKG2A axis, which then here really translates into a peptide-specific NK cell recognition of NKG2A expressing cells. And we propose, based on these results, that viral adaptation to NK cell responses may drive a strain-specific recognition by NK cells, depending on which peptide is present. And just as a brief future outline, we are really interested in further studying NK cell-mediated selection pressure and the evolution of viral strains. And for this, we return to an old friend, which is cytomegalovirus, and we do that with a new friend, which with Rich Stanton from Cardiff University. Based on our previous data, we know that there is a lot of diversity in terms of different variants and different strains in cytomegalovirus, but also the literature really shows us that mixed infections in CMV cases in patients is not common. It's not rare. That is a common occurrence. In around 1/3 of all patients, you can detect 2 or more different virus strains simultaneously. And one can also follow this diversity in individual patients. So one example of intrapatient dynamics that we observed is here on the right side, where shortly after transplantation in this patient, there was a CMV reactivation where we could detect 2 variants, one orange, one violet, of CMV. And then around 100 days later, there was another secondary infection or reactivation, which then only in which only one of the 2 variants was detectable. And we are now developing in vitro assays to study variant selection, where we mix target cells that represent different CMV variants together with NK cells, and we can clearly distinguish and track them. And then depending on which phenotype of NK cells, so either from different donors or even different subsets within one donor we add, we can either find no effect at all on variant selection, or we can, in the latter case, phenocopy the selection of the orange CMV variant that we had observed in the patient. And I think this workflow will really provide us with a toolbox to, in the future, study the molecular mechanism, the functional requirements, and also the phenotypic features that can be involved in NK cell-mediated immune pressure and how it affects virus evolution. And with that, I would like to thank all the people involved, of course, the members of my team, especially Eleni, who was driving a lot of the experiments, but also our collaborators locally and internationally. Really appreciate their contributions. And then I'd like to close with kind of a short personal update that in autumn this year, we will move the center of our activities from the Karolinska Institute in Stockholm to the University of Kiel in Germany. So if you're interested and excited about our work and would like to work on that with us in this institute for immunology in Kiel, a city that's at a beautiful part of the coastline of the German Baltic Sea, then feel free to get in touch with us. And thanks for joining today.